Hello friends and welcome. Today we will be discussing natural pigments of plant and animal origin and their significances. As we know that pigments are chemical compounds that absorb light in the wavelength range of the visible region. Pigments can be classified by their origin as natural, synthetic or inorganic. Natural pigments are produced by living organisms such as plants, animals, fungi and microorganisms. Synthetic pigments are obtained from laboratories. Natural and synthetic pigments are organic compounds. Inorganic pigments can be found in nature or reproduced by synthesis. Pigments are natural substances in cells and tissues of plants and animals that impart color. Many food pigments are unfortunately unstable during processing and storage. Prevention of undesirable changes is usually difficult or impossible. Depending on the pigment, stability is influenced by factors such as the presence or absence of light, oxygen, heavy metals and oxidizing or reducing agents, temperature and water activity, pH, etc. Because of the instability of the pigments, colorants are sometimes added to foods. Now we will discuss about pigments of plant and animal origin. First, heme compounds. Heme pigments are responsible for the color of meat. Myoglobin is the primary pigment and hemoglobin, the pigment of blood, is of secondary importance. Most of the hemoglobin is removed when animals are slaughtered and bled. Thus, in properly bled muscle tissue, myoglobin is responsible for 90% or more of the pigmentation. The myoglobin quantity varies considerably among muscle tissues and is influenced by species, age, sex and physical activity. For example, pale colored veal has lower myoglobin content than red colored beef. Other minor pigments present in muscle tissues include the cytochrome enzymes, flavins and vitamin B12. Now we will discuss about myoglobin or hemoglobin. Myoglobin is a globular protein consisting of a polypeptide chain. Its molecular mass is 16.8 kilodalton and is comprised of 153 amino acids. This protein portion of the molecule is known as globin. The chromophore component responsible for light absorption and color is a porphyrin known as heme. Within the porphyrin ring, a centrally located iron atom is complexed with 4 tetrapyrrole nitrogen atoms. Thus, we can say that myoglobin is a complex of globin and heme. The centrally located iron atom shown possesses 6 coordination sites, 4 of which are occupied by the nitrogen atom within the tetrapyrrole ring. Hemoglobin consists of 4 myoglobin linked together as a tetramer. Hemoglobin, a component of the red blood cells, forms reversible complexes with oxygen in the lungs. This complex is distributed via the blood to various tissues throughout the animal where oxygen is absorbed. It's the heme group that binds to molecular oxygen. Myoglobin within the cellular tissue acts in a similar fashion, that is by accepting the oxygen carried by the hemoglobin. Myoglobin thus stores oxygen within the tissues, making it available for metabolism. Meat color is determined by the chemistry of myoglobin, its state of oxidation, type of ligands bound to heme and state of globin protein. An important means of stabilizing the meat color is to store it under appropriate atmospheric conditions. The problem of discoloration is caused by heme oxidation, that is when ferrous atom change to ferric atom, and it can be resolved by using modified atmosphere packaging. This technique requires the use of packaging films with low gas permeabilities. After packaging, air is evacuated from the package and the storage gas is injected. By employing the oxygen enriched or devoid atmosphere, color stability of meat can be enhanced. Second pigment of plant origin is the chlorophyll. As we all of us know that chlorophylls are the major light harvesting pigments in green plants, algae and photosynthetic bacteria. They are magnesium complexes derived from porphyrin. Porphyrin is a fully unsaturated macrocyclic structure that contains 4 pyrrole rings linked by single bridging carbons. Pyrrole carbon atoms on the periphery of the porphyrin structure are numbered 1 through 8. Chlorophylls are located in the lamellae of intercellular organelles of green plants known as chloroplasts. They are associated with carotenoids, lipids and lipoproteins. Weak linkages, that is non-covalent bonds, exist between these molecules. The bonds are easily broken, hence chlorophylls can be extracted by macerating plant tissues in organic solvents. It's well known that chlorophyll content in plants vary greatly depending upon the metabolic processes that are reliant primarily on physiological, genetic and biochemical factors, but also on external influences, that is the climatic factors, the agricultural practices, season, geographic growing region, post-harvest handling, storage and plant part considered. There are pronounced differences in chlorophyll contents among stems, leaves, peels and pulps of vegetables and fruits. In general, it can be stated that higher chlorophyll contents are mainly found in leaves, and the more colored they are, the higher amount of chlorophyll they have. Therefore, we can say that the highest amount, as much as 1.5 to 2% fresh weight, can be found in fully developed leaves of spinach, parsley, kale and green cabbage, for example. High performance liquid chromatography, that is HPLC, today is the method of choice for separating individual chlorophylls and their derivatives. The chlorophylls can be altered by various methods like enzymatic treatments, heat and acid treatments, metallocomplex formations and allomerization. Next, we will discuss about carotenoids. Carotenoids are the nature's most widespread pigments, with the Earth's annual biomass production estimated at 100 million tons. A large majority of these pigments are biosynthesized by the ocean algae population. In higher plants, carotenoids in chloroplasts are often masked by the more dominant chlorophyll pigments. In the autumn season, when chloroplasts decompose during plant senescence, the yellow-orange color of carotenoids become evident. In all chlorophyll containing tissues, carotenoids function as secondary pigments in harvesting light energy. The photoproduction role of carotenoids stems from their ability to quench and inactivate the reactive oxygen species formed by exposure to light and air. Carotenoids are comprised of two structural groups, the hydrocarbon carotenes and the oxygenated xanthophylls. Oxygenated carotenoids that is xanthophylls, consists of a variety of derivatives frequently containing hydroxyl, epoxy, aldehyde and keto group. Over 560 carotenoid structures have been identified and compiled. The basic carotenoid structural backbone consists of isoprene units linked covalently in either a head-to-tail or a tail-to-tail fashion to create symmetrical molecule. Other carotenoids are derived from this primary structure of 40 carbons. The most common carotenoid found in plant tissues is beta-carotene. This carotenoid is also used as a colorant in foods. Both the naturally derived and synthetic forms can be added to food products. Some carotenoids found in plants include capsanthin, lutein, it's a diol of alpha-carotene, its esters, marigold petals and bixin. Other common carotenoids found in foods include zeaxanthin, that is a diol of alpha-carotene, violaxanthin, an epoxide carotenoid, neoxanthin, an alanic triol, and alpha-cryptoxanthin, that is a hydroxylated derivative of alpha-carotene. All classes of carotenoids, that is hydrocarbons, carotenes, lycopenes and oxygenated xanthophylls, are lipophilic compounds and thus are soluble in oils and organic solvents. They are moderately heat stable and are subject to loss of color by oxidation. Carotenoids can be easily isomerized by heat, acid or light. Since they range in color from yellow to red, detection wavelengths for monitoring carotenoids typically range from approximately 430 to 480 nanometers. Next, we will be discussing about flavonoids and other phenols. First, anthocyanins. Anthocyanins are responsible for a wide range of colors in plants including blue, purple, violet, magenta, red and orange. The word anthocyanin is derived from two Greek words, anthos, which means flower, and kyanos, which means blue. These compounds have attracted the attention of chemists for years. Two of the most notable investigators being Sir Robert Robinson and Professor Richard Willstatter, both were awarded Nobel Prizes in chemistry in part of their work with plant pigments. Anthocyanin pigments are relatively unstable, with greatest stability occurring under acidic conditions. Both the hue of the pigment and its stability are greatly impacted by substituents on the aglycon. Degradation of anthocyanins occurs not only during extraction from plant tissues, but also during processing and storage of the food tissues. Anthocyanins are the most prevalent flavonoids. Although yellow color in foods are attributable to the presence of carotenoids, some are attributable to the presence of non-anthocyanin type flavonoids. Next, we will be discussing about other flavonoids. Non-anthocyanin flavonoids make some contribution to color in foods. However, the paleness of most non-anthocyanin flavonoids generally restrict their overall contribution. The whiteness of vegetables such as cauliflower, onion and potato is attributable largely to non-anthocyanin flavonoids, but their contribution to color through co-pigmentation is more important. The chelation characteristics can be converted to colored products during food processing. Pro Anthocyanidins are also referred to as leucoanthocyanidins or leucoanthocyanins. Other terms that are used to describe these colorless compounds are anthoxanthin, anthocyaninogens, flavolins, flavolins, flavonogens. Now we will be discussing about betalains. Plants contain betalains have colors similar to plants containing anthocyanins. Betalains are a group of pigments containing beta-cyanins, that is red in color, and beta-xanthin, yellow in color, and their color is not affected by pH, contrary to the behavior of anthocyanins. They are water soluble and exist as internal salts, that is zwitterines, in the vacuoles of plant cells. The presence of betalains in plants is mutually exclusive of the occurrence of anthocyanins. Betalains are vacuolar plant pigments. Although they are slightly soluble in ethanol and methanol, water is the best suited solvent both for stability and solubility reasons. In contrast to anthocyanins, the betalains are even more polar, as can be demonstrated by shorter retention times in reverse phase high performance liquid chromatography and lower solubilities in alcoholic solutions. Betalains are considered to be stable at near neutral conditions in foods that are devoid of sulfites, protected from oxygen and light, and stored for short times at cooling temperatures. Hence, typical food commodities colored with betalains include dairy products, fruit fillings for bakery products, relishes, various instant products, confectionery, meat substitutes, and sausages. Now we will be discussing about quinones. Quinones constitute a large group of natural pigments which are widely distributed in higher plants but also are present in various fungi, lichens, and some invertebrate animals. The common structural feature of quinones is the presence of two ketone groups on a monocyclic or polycyclic aromatic skeleton. Quinones cover a wide range of colors from pale yellow to orange, red, purple, and brown. Quinones are among the most widespread pigments in nature, characterized by great structural diversity. Ubiquinones are ubiquitous molecules. Plastoquinone is located in the chloroplasts. Menaquinone is produced by the microorganisms and phylloquinone by plants. In higher plants, quinones can be found in leaves, flowers, fruits, roots, bark, and heartwood. There is a long history of utilization of quinones in dyes. The best known example is the Lawson naphthaquinone, which is present in the henna, a natural dye obtained by crushing the leaves of Lawsonia alba. Another well-known naphthaquinone is juglone, isolated from buds, nut hulls, and roots of walnut trees, that is Juglans regia. Juglone can also occur in glycosylated form, that is hydrajuglone alpha-D glucopyranoside, representing 6 to 8% dry weight in young organs. Quinones play an important role in the coloration of some fungi, lichens, insects, and echinoderms, but they really contribute to the external colors of higher plants. Ubiquinone, which is also known as the coenzyme Q, plays a crucial role as a respiratory chain electron carrier transport in inner mitochondrial membranes. It exerts this function through its reversible reduction to semiquinone or to a fully hydrogenated ubiquinol, accepting 2 protons and 2 electrons. Because it is a small lipophilic molecule, it's freely diffusible within the inner mitochondrial membranes. Ubiquinones also act as important lipophilic endogenous antioxidants and have other functions of great importance for cellular metabolism. Plastoquinone in plant chloroplasts and menaquinone in bacteria play similar roles in electron transport. Phylloquinone, that's vitamin K1, and menaquinone, that is vitamin K2, are essential for humans. They function mainly as lipid cofactors in blood clotting. Next, we will be discussing about the animal pigment, that is melanins. Melanins are complex polymeric structures which are usually mixtures of macromolecules. Melanins are produced in mammals in 2 types of cells of different developmental origin. First, in the melanocytes of the skin, hair, choroid and iris, and second, in the retinal pigment epithelium. Specialized organelles of the melanocyte, that is the melanosome, synthesize and store eumelanins and pheomelanins. Melanin biosynthesis in animals is a complex process starting with the L-tyrosine amino acid. In the first step, L-tyrosine is converted first into dopa and then into dopaquinone, a process catalyzed by tyrosinase. In the biosynthesis of eumelanins, dopaquinone undergoes a cyclization to form dopachrome and subsequently a tautomerization into 5,6-dihydroxyindole 2-carboxylic acid. Melanins are classified as eumelanins, pheomelanins and allomelanins. Eumelanins are produced mainly by animals, but sometimes they also occur in plants. Eumelanins provide black and brown color of animals and can be found in the granules in hair, feather, fur, skin, scales and integuments. They are also produced in inner tissues such as choroid, iris and inner ear. Now let us talk about the functions of melanins. The most evident function of melanins is providing pigmentation to mammals, invertebrates and plants as well. In animals, melanin pigmentation is important for camouflage and display and even in defense, as in the case of cuttlefish ink. In invertebrates, melanin is involved in immunity, wound healing and sclerotization, that is cuticular hardening. Accumulation of the melanin in the skin was considered as a protective response to the damage induced by ultraviolet radiations. It seems that melanin protects cells by absorbing the photodamaging light, blue light and scavenging the reactive oxygen species. It seems that melanin protects cells by absorbing the photodamaging blue light and scavenging the reactive oxygen species. Melanins can bind toxic metals and this property may have a role in antioxidant defense and in transcutaneous metal excretion. The role of allomelanins is not completely understood, but it seems that their accumulation is related to the maturation of truffles. Plant melanins show antioxidant, liver protecting and anti-tumor properties when tested in various experimental models. Recently, it was shown that fungal melanins are linked to virulence in some human pathogenic and phytopathogenic fungi, and they protect fungal cells. At last, we'll conclude our today's topic by saying that pigments are present in all living matter and provide attractive colors and play basic roles in the development of organisms. Human beings, like most animals, come in contact with various surroundings through colors, and things can or cannot be acceptable based on their color characteristics. The basic information about natural pigments of plant origin is focusing attention on carotenoids, anthocyanins and betalains. Special considerations are given to their salient characteristics, that is, to their biosynthesis, taking into account the biochemical and molecular biology, information generated in their elucidation, and to the processing and stability properties of these compounds as food colorants. That's all from today's lecture. Hope you have understood it well. Thank you very much.